Hi, my name is Quincy Williams, and this is a presentation on the effect of muscle fatigue on motor unit recruitment during isometric contraction. Multiple studies have shown that during prolonged isometric contraction, motor unit recruitment increases as the muscles fatigue. This phenomenon can be assessed using electromyography or EMG. EMG has been shown to be a consistent and reliable way to measure the neurological response of muscle contraction. The purpose of this study was to investigate the relationship between motor unit recruitment, muscle fatigue, and time using EMG on wrist flexors and extensors. We hypothesized that our results would match that of the literature and that over time, motor unit recruitment would increase. In order to conduct this study, 8 students from the DPT class of 2021 were chosen to participate. 5 male and 3 female subjects were chosen with a mean age of 24.5. Inclusion criteria were that the subject was a current member of the 2021 DPT class and had no previous condition that resulted in injury or weakness to wrist flexors and extensors. 6 EMG electrodes were placed on the wrist flexors and extensors on the dominant hand of the participant, as seen in Figure 1. The subject held the dynamometer with the wrist in neutral position, and the grip was adjusted to the individual's hand. Peak EMG was measured by having the subject squeeze the dynamometer as hard as they could, and the highest value was recorded. Then 25% of the maximum was calculated, and the subject held the dynamometer at that level until fatigue, or 300 seconds. EMG values for both wrist flexors and extensors were recorded every 15 seconds. All data was normalized as a percentage of maximum EMG, and the mean and standard deviation for all subjects was compiled and graphed, as seen in Figures 2 and 3. Figures 2 and 3 show that overall, there was not a significant increase in motor unit recruitment over time. There appears to be a slight upward trend in wrist flexors, but the large error bars do not make them statistically significant. Overall, motor unit recruitment in wrist flexors and extensors maintained relatively static over time in our study. Our initial hypothesis was not supported, despite the amount of evidence found in the literature. There were several sources of error present that likely caused this variation in our data. First, there was a high degree of user error as the individuals conducting the study had not used EMG very much prior to this study. The electrode placement may have been inconsistent among the 8 participants. Finally, and most importantly, there was variation between the time to fatigue among the 8 participants. Also, the participants had varying degrees of muscle strength in wrist flexors and extensors. Normalizing the data combats this to a certain extent, but it cannot completely eliminate the effect, and this is where the majority of error likely comes into play. This research is clinically relevant because it is important to know the neurological mechanisms behind muscle fatigue to educate patients to help minimize injury risk. Knowing how fatigue works helps physical therapists appropriately prescribe exercise for the individual patient. Thank you all for listening, and I'd be happy to answer any questions.